strange to be up here as an editor. So I've been here in 2011 as a young investigator, some of you in your shoes. And then I came to the 10th year of YIM, and it was more like a reunion of all young investigators. And this one, so my lab actually works in mitochondrial protein synthesis, and we think of the mitochondria as the rheostat of the cell, where you can dial it up, dial it down, depending on what the cellular needs are. Right? So before I show any work, I just thought I'd give a brief overview of where I started from. I did a bachelor's in chemistry at Delhi University, and then at the end of my 3 years, I wanted to get out of chemistry and move towards biology. Right? It was very difficult at that point to transition of it. But IIT Bombay, right, in all its wisdom at that time, used to only have an entrance exam for chemistry students. Right? So our class was full of chemists trying to do biology. And that's where I went for my master's. This is my thesis supervisor. And then once I was done here, I somehow moved to University of Michigan, where I did a PhD with Nadar. And then I did a postdoc in Larry's lab for 4.5 years, 5 years. Right? So once I was done with that, I moved to Delhi University, South Campus, where I was working. So what's the mitochondria known for? I'll get into how I got here. But the best thing that we remember from the textbook is it's required for ATP. So if I could have a show of hands of how many people are aware of the fact that mitochondria has its own DNA. Oh, wow, that's great. Because at the end of my thesis, there was this well-known microbiologist, this was around 15 years back or 20 years back, where he said, I almost forgotten mitochondria. So, what does the DNA encode for? It actually is very critical for almost all organisms that uses oxidative phosphorylation. It encodes most of the times for the subunits of various complexes that are a part of the mitochondrial DNA, as well as a part of this ribosome machinery that is unique to the mitochondrial system. Here, the proteins are encoded in the nucleus, we know that they come in and they assemble here. So, how did so when we think of the translation system, we remember prokaryotic translation, we study this, and then a lot of focus goes on to eukaryotic translation. So when I started my lab, very little was known about translation systems in the mitochondria. So here's a couple of examples. So the mitochondrial translation, the mammalian mitochondria, that's us, actually doesn't have a UTR at all. Right? There's only 2 or 3 nucleotides. Whereas yeast, which is the model organism that I work with, has a very large UTR before you start off with potency. Right? And so when the people known about the fact that there are these mRNA recognition complexes, and somehow 37S subunit, the small subunit joins, how does it know it doesn't know? And does it scan and how does it recognize an agent? It's still not known how this happens in mammalian cells. There's a small and a large subunits, magically they come together and start translation. So before I show you what we work on, so how did I get here? So my model organism is Saccharomyces cerevisiae, so it's a baker's yeast. It's well known for making beer and bread and things like that. And I was in the lab as a master's thesis. And so this actually talks about the role of mentors. And he was a fantastic geneticist, right? Very down-to-earth teacher. And he was looking at galactose utilization. So this is what got me interested in Saccharomyces. When I was starting my PhD in Janine's lab, Janine was not a yeast biologist. She was actually a microbiologist. And she was working with colobacter placentas and looking at differentiation in that model. What her lab had done at that point of time was purify this protein, OBGs and GTPase, and it was early days in the GTPase field, small molecular GTPase field, or fairly early days, where RAS had been discovered, and RAS was so important for cell growth that it was the causative form of cancer. But this GTPase had a completely different kinetics. Right? It didn't bind nucleotides or anything. So I came into the lab and I said, Janine, can you work on the yeast homologs of it? Right? So me and a couple of other grad students started working on this OBG family of GTPase. It was used to be labeled as with its, it was an unnamed gene, unknown gene, no function was assigned. Over the years in her lab, we figured out that OBG is required for a late step in bacterial ribosome biogenesis, and my thesis was on MTG2, which was the mitochondrial membrane. So once it was done, I went to Larry's lab at Scripps, and if you remember the talk from the first day, at Scripps, you're made to write a lot of independent grants, right? As a postdoc, you are required to generate your own funds, right? So you're never supposed to, right? And I wrote 3 grants, and all of them were rejected. Right? So sometimes failure can be good for you. Right? Even though I failed in writing the grants, but the fact that I had to write 3 times, 3 different grants, it taught me how to split up a problem into small parcels of questions, right? And the other thing that it does, it motivates you to go and look for a job outside your postdoc lab. So that's what it really motivates you to get out of there, get out of this up or out funding situation. So this is 2008-9. We didn't have YIM. I think the first YIM was somewhere in Boston, I think 8 or something. But we had an informal group of Indian postdocs, and we saw this advertisement in Science for faculty positions at Delhi University. I wasn't completely ready to apply for a job, but this rejection of grants forced me to, I was like, okay, let me apply and see. So I got in. And then I was like, okay, I have a faculty job, but what am I going to work on? I didn't want to work on what I was doing in my postdoc. My postdoc mentor wasn't very happy with the fact that I was leaving. But he was okay. Once I told him that I really wanted to do it, he was fine. So I went back and I looked at what was the training in Janine's lab. And what Janine had made me do as a part of my thesis was go and look. So she was a mitochondrial person. Go and look and see what all proteins might be required for ribosome function, right, bioinformatically based on what was there out of the literature. So I had all these genes that I thought were important for mitochondrial functions, and they're spread out here. So when I started my lab, I thought I'll just write a grant on all of them together under one umbrella, mitochondrial function. So when I moved back, it was early days, and the DBT Wellcome Trust Alliance had just formed. I knew some people that had successfully applied for the Wellcome Trust when it was straight from Wellcome Trust. So I applied. Obviously, we got rejected at the university. They didn't want my proposal, which was okay, fine. So then I did was, I parceled these out into DBT, CSIR, 2 grants to DBT, 1 grant to CSIR, and I got my lab started. So that brings to what we work on. So what we ended up doing was we looked at mitochondrial translation, and shown here is how COX-1, which is a part of complex 4, exceptionally important protein. It's where the heme binds, and complex 4 is where the oxygen, the last electrons are transferred to the oxygen. So if you don't assemble this thing correctly, you're going to end up generating a lot of reactive oxygen species and you're going to die. here itself. So there's a whole bunch of literature that's in here. So what my lab has shown is that MRXA is a GPase that's required for translation of COX-1 under stress conditions of cold, and a couple of other stress conditions I won't get into that. But what we've also looked at, so this is some unpublished, published work. So this is a great example of where the reviewers don't like your entire story, so you better split it into a smaller paper, keep your lab going. So here we're just showing that it actually binds to the ribosome via the C terminus. And this is the data that shows that it's required for loading of mRNA. So you have a gene that's supposed to be requires for loading of mRNA on the 37 subunits. And of course, this is conserved between humans and yeast, whereas all the other stuff that I talked about in the beginning of mRNA recognition complex is broken. So if you remember, I said that mammalian cells, there's no nucleus. Okay, fantastic. Then we come to the third project, and this is where we started thinking of, is there a way that mitochondrial translation is regulated? So this is a wonderful clip that a student of mine had come up with. What she's showing is that can we have RNA structures within the mitochondrial mRNA coding sequence? Is there a way that they form structures which are dynamic in nature? And then there's a protein that opens up. That's that. Okay. So can there be a stimuli which leads to highly structured? So Saccharomyces cerevisiae is a great model because you actually can control the amount of mitochondrial oxidative phosphorylation. You just grow them in glucose. It's kept at a very basal level and move them to glycerol. So this idea comes from this paper or this project that this grad student did over a long period of time, where she was looking at this RNA helicase, and what she saw was there was a dramatic shift in the level of control on mitochondrial translation products if you just grow the cells in glucose, or if you should grow the cells in galactose, under conditions of which there is a whole body of literature which talks about how much of the oxidative phosphorylation happens. Right? So we published this in MCD. She went off to Yale for a postdoc. And she left us with this model. Right? So our model was, that either IRC3 works in tandem with different proteins to resolve the structure, or there are different structures within the coding sequence that it resolves. So she'd shown that she did a lot of work to show that's required for translation information. And this is something that's been at an approval stage, so it's been approved in principle by ANRF. We still haven't had money. So this talks about what kind of work you do. Right? So, this is for the young PIs. When you choose to apply for a job, or not young PIs, young PDs, to be young PIs, it's really important to figure out a biological question. Right? That's something in your hand. See what you actually enjoy. So I picked yeast, and I picked mitochondrial function, and I knew that I could do this in cheap. Will I get funding or not? I have no idea. When you're a PDA, you're a young PI, you just come, you have no idea if you're going to be able to raise any grants, but you know that you'll be able to get some money somewhere. There's always some hope for it. Can we do it a bit cheap? And I actually had an experience in that because when I was in Janine's lab at University of Michigan, I was on an unfunded project. I taught my way through grad school. That's where my stipend came. But we were able to use whatever resources was in the lab to build the project. Right? And she was quite happy you take it. She was more than thrilled that you've gone off to do a postdoc at a much better funded place, and you want to come back and work on something that you started with. Okay. So now, here comes the vagaries of a university or any institute in India. Funding is dependent on institutional support. Institutional supports vary, especially if you come to a university, depending on who the leader is. So a university is not always led by a scientist. So when I came, we had a very dynamic vice chancellor, Professor Deepak, a well-known plant breeder and a biotechnologist in the country. So under his leadership, the university was giving a startup grant of 4 lakhs. I thought that was a lot of money. And then when I moved back, I realized it's only $10,000. So what can you get with it? But that was okay. But at least $10,000 is good enough. But that rupee was just money. There was an princely sum of R&D support of 2.5 lakhs. There was a decent amount of conference support for PI as well as students. And the administration was very kind to scientists because, see, biologists, we had to do a lot of purchases. We were purchasing equipment, we were purchasing reagents all the time. Right? So if you don't have an admin support, or you can't get the admin to support you, you're in a painful situation. So he retired in 2010 August. I joined just before that. So, but he moved the machinery so much that these things lasted for a little bit, even after he left. But it took only 4 or 5 years for R&D support to completely dry out. Right? So you don't have any consumable internal money. The startup grants go away. Conference support became more difficult. And the worst was the administrative support, so came down. Okay? So when you are at a university, you've got to make sure that you can't control this, you can't control this, but you can control some of the administrative support. You need to go and talk to your finance officer, you need to go talk to the person that's there. Make sure that you understand all the rules. Right? So where do we get funds? This is something that Sanjay Mishra showed in the beginning. I've modified the data from DSD. This is where DRDO stands. These are the agencies that we usually apply to. CSIR, DSD, DBT, ICMR. And that's the total cost. Where are we competing? So this is the amount of money that extramural research support is there in India. So there's DSD, that's the fraction that's in DBT, fraction that's in ICMR, and CSIR. And this is data from 2019. A couple of year old data, but that's all that's publicly available. So you have to be, as a PI at a university, you need to figure out how to parcel your stuff into multiple small projects, and you have to hit all the grant deadlines of it. There's just no way around it. At times you have to maybe publish something that doesn't give you a high impact paper, just so you show productivity. And we've been fairly good about it. So we've got some fundings from SERV, a couple of projects from SERV, a couple of projects from DBT. I even applied to BRS. This was a DAE organization. They haven't come out with a call for the last few years, but I'm hoping that they will come. ICMR used to have a great support for students. Right? So if you have a student that's about to finish with their funding, but would like to stay back and finish a paper, right? ICMR had an SRF program that gives students SRF RA program. So ICMR has it, and so does CSIR. So hit them up. So as a PI, it's your responsibility to make sure your student at least hits all the places where money can be found. So plan your funds, figure out the competitive grant systems, and then plan your funds such that you can get collaborative grants within this. So when I came, I needed an ultracentrifuge. So I put this in a DBT grant, which was itself for 13 lakhs. But then it wouldn't work in any of the centrifuges that were there on campus. So I went and convinced. Okay. Sensitive. I'm almost done. So we needed this ultra. So I convinced a lot of my colleagues to apply for these institutional grants that Sanjay Mishra was talking about. We applied for a FIST fairly early on, so I started in 2010. By 2013, we got the money, we got the stuff running, and it helped the lab. And of course, when you convince others, there's lots of equipment that come into the department. So here's the take-home message. Chase the funding agency, chase wherever the money is. And for young PIs, read the purchase rule books. There's lots of loopholes in Indian system. Figure out what the loopholes are, get the damn admins to learn what the loopholes are and convince them. And you'll be able to get your, you'll be able to spend your money and won't have to complain about jet purchases and things like that. Use the flexibility to your advantage. Use your colleagues. So this is some of the people that are working with me. And at a university, you're unlikely to find buddies that work with you on the same project or near about the same project. So it's a wonderful place to actually talk to people that are not in your field, but you've got to work together. Build your research, build your MSCs. Finally, is this hard work, is it worth running a lab? And a lot of times you feel, am I doing this correctly? And so here's an email that I got some time back from a master's student who was thanking me for giving some advice. And it was a very funny thing because she was this really bright student who comes into my office after an EMBL interview visit, and she said, "Am I even good enough to do a PhD?" I was like, you're being foolish. Go away. And then obviously I never heard back from her till she graduated, and I'm sure she's doing really well. I have no idea where she is. Count your small victories and don't forget to have a good time. This is my group over the years and in my lab. Thank you very much for your attention. And I'll take over.
Thanks.